Hello, hi, and I'm back before you. This time it's the same thing that I did the last time, making of a dipeptide with the same amino acids, glutamine, arginine, and lysine. And I would be looking at an enzyme. You see, I did organometallic catalysis, and I thought this time around I would try an enzymatic catalysis to make the same dipeptide. And peptidyl transferase affects this. Peptidyl transferase, as the internet said, as Google said, or the Google AI overview said, is present in the ribosomes of the RNA of plants. I mean, the cells of plants, but in the RNA, present in the RNA of plant cells, of course, plant cells. And I thought I could use parsley, edible, right? I mean, I'm using the micropipette tray again, and so parsley flakes. And but then somebody got into my head and said fresh parsley would have more RNA, would have more RNA, and would have more RNA. So hence the reaction would be instantaneous, absolutely instantaneous. So three substrate concentrations, and I'm just going to move the computer to this side so you can watch me. Strands of hair make me nervous. Although I am a hair lover because I have hair, but uh, you know, flowers. As engineers, flowers. Once in a while, maybe to give the odd Professor Phillips of rose, but I never did. I never did. I think I after graduating my Master of Engineering degree, I gave her a box of chocolates, small box of chocolates, as I did my supervisor, Bradley Isabel, I think. If I remember correctly. I could be wrong on this, or I think I got pregnant, something, or I've made it into MASC and I gave Professor Phillips, who was the graduate coordinator then. But, you know, um, because she wouldn't come out, maybe I invited her for a coffee and she wouldn't come. So Jane Phillips. So to give, or Professor Chang for that matter, hair again. All right. Less of, okay. Flowers, hair make me nervous. All right, because engineering is all numbers, facts and numbers and analysis and that sort of thing. So again, the micropipette tray, 30 ml of water in each, so pH 7, and I'm adding pressure here. Exhaust is on, just in case there's ammonia being released or carbon dioxide. And so I begin again. All right, so one, two, three. So substrate concentration, one of each amino acid. So 500 milligrams. So one of each amino acid, one, or or maybe we do one here, and two here, and we have one, three, and one, two. The third one's going to be tough because it has nine capsules in it. Three, one, two, three, last one, two. I'm blocking the view. Two. Just putting in these capsules. You can somehow get back to me, maybe through the university, and let me know if you did the mass transfer fixed law of diffusion. Um, the okay, this might be good. One. Fixed law of diffusion of a capsule. I just concentrate on this. Oh, I did something wrong. One. Okay. No more adding. No, this is good. This is good. This is correct. The other two, dipeptide. So two more, two more. So S1, S2, S3. Substrate concentration 1, substrate concentration 2, substrate concentration 3. So lysine, last lysine. Oh, one, 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 two, two, two. Maybe I should take it 21 mil, 25 mil. Two, two, and lysine 3, 3, 3. Uh-oh. I've used up lysine. Lysine, I only get 3 points. There's only one more remaining. And then it's set. Lysine ran out. I ran out of amino acids. So three right points, and then we'll see if S2 and 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 4 and 5 of S3, which is not S3 actually. I don't have lysine at all. So S2, so this would be, what would this be? This would be, is that arginine? So it's all right. Arginine, 99. All right, it's all good. So this, I'm looking at the confusing ones which don't have the right number of lysine there. All right. So those two are, no 2 points. So S1, S2, S3 for 3 points only. All right. So I'm beginning the experiment. Stir, stir, stir. Experiment. So timer. Let's get this set. This set. Okay. Okay. Enzyme goes in. Enzyme, peptidyl transferase. So, it's hard to get. I crushed this in the mortar and pestle, chopped it, crushed it. So there's enzyme, and this is a bit much. Enzyme. Maybe a little bit with the fingers. They may be easier. Enzymes we have. Enzyme. 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 It's easier. Somebody would think this is some kind of engineering potion. It's going to make it all work. And it is. Enzymes are the key. All right. Stir, stir, stir. The other two, I'm going to let it go. So this remains. Just going to wash my hands. And fresh parsley. Paisley. I think there was a model, Paisley. Models have no. All right, now I'm going to begin the reaction. Stir, stir, stir. All right, key to it. Micro mixing, macro mixing. Stir, stir, stir. Stir, stir, stir. Stir, stir, stir. Stir, stir, stir. Stir up a reaction. And then this is 3 points only. Stir, stir, stir. Stir, stir, stir. I'm supposed to hand this in by 3. Sign from heaven. The chair this time. Christopher North in the University of Toronto, Department of Chemistry. Yeah. So what's the time? 1 more minute. We'll stir, stir, stir. Stir, stir, stir. Stir, stir, stir. Okay. Okay. Right time, first point. Okay. One third. One third. Okay. And it's the same amount. I could have the same results. Same amount of enzyme, twice the substrate concentration. I think. So this is 1/5, I think. 25 by 3, 25 by 5. That was my lucky measuring cup. This is, it's always optimum enzyme loading, right? Uh, just, stir, stir, stir. Blocking the view, I'm coming. Stir, stir, stir. So there's a ribosomal DNA, RNA, and then mitochondrial. Minutes, stir, stir, stir. Take the lucky cup. Yeah. So and then so the gradations are 25. This is again 25 by 3. Twenty-five by 2. Different paste. gloves never do this, but this is because it's amino acid and enzyme and 25 by 3. Okay, next, stir, stir, stir. Enzyme kinetics. Okay. 6, when the marker is 6. Six. Okay, I'm going to take the samples. Some 25 by 2. By 2. It's all the same substrate. It's just different concentrations of substrate. Oh, this I think has become completely ammonia and carbon dioxide. 95, 25 by 6. Okay. Stir. Completely stirring, 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 stirring. It's not a neat linear curve in a way because it plateaus off. Enzymatic reactions go this way, plateau. So dP versus dt, the product versus time gives the V naught, velocity of reaction. And from the plot of different substrate concentrations versus varying V naughts, you get the Km, Michaelis-Menten constant, and the Vmax. Uh, 3 and setting 3. Setting 3. So does it indicate anything? White, you can tell it's the same amount of enzyme. And what's the time now? It's all right. It's the right time. 5 by 2. So I have no point for 6. Last point in. Once the finger goes off. So you don't get it? Well, wait. So we used to talk. Listen to the music. I should turn the music on for luck. For love luck. All right. Classic Q107. It's completely unprofessional if a professor walks in. All right. But our professor was quite okay. Become a really strange man now. But, back then, I think he always used to make us work a lot and, see to it that we did varying things, varied our experiments, and played a little bit of foul here and there. Oh my God! Stir, 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 stir. Okay, I'm taking it down. 25 by 5. 25 by 3. And that concludes my experiment. All right. So what do I have here? So I think the timer sometimes, measures more. Remember my exercising? I'm exercising all those experiments with exercises, exercising and exercising and exercising, and the timer is not going off. My watch is showing the right time. So there we go. So here are my results. So, point for each amino acid, point 0.5 plus 1 plus 1 and 1.5. And so you have 25 by 3. So product. So I would have to take, water minus 1 teaspoon of water. The volume has to be same. The mass is different, of course. So the volume was the same, and maybe density of mixture study or plain. No, this is just actually plain. Um, just subtraction. From the concentrations that would give me a product, and it's if it's stinging again, it's ammonia. That's what I'm told. I have not confirmed it, and but then turned a little red. I thought, oh, that could be, yeah, but fertilizer, right? Everybody uses ammonia and that sort of thing, and the hair color has ammonia, had ammonia. Now it's all changed. Uh, so, um, so this concludes my experiment for the day. I showed you my results. Um, so, as I said, you could calculate the velocity of reaction, and then so product would be. I would have to assume some, what I get from just plain subtraction of the original amount minus is all product. So I may not have made as much of dipeptide as I would like to have, because some of it maybe with enzymes there's too much reaction, very fast, very instantaneous, very quick. So it may have just so happened that I have made carbon dioxide and the ammonia as well, and the bonds eventually broke up. So the correct place to stop the experiment is, you would have to have add an inhibitor, I guess, to the entire reaction, to the enzyme, and that would conclude the reaction. Yeah, that would conclude the reaction. All right, signing off. Thank you, Gayatri BC Therapy.